in theoreticians community, they always make some jokes about experimentalists.
So what is the joke?
Why do you crystallize them? I mean, why they are important?
Because we want to get as close as possible to your theoretical ideas. We want to make the world, well ordered. And in our case, we are looking after exciton diffusion in organic semiconductors. And exciton diffusion actually is more pronounced in crystallized materials. Because we are surface scientists, so we put our material on the surface because we care about the surface. I don't know, then I will be doing, I don't know, I would become a DJ then. I would have no idea about this stuff. And also in AFM, we are using a cantilever that measures the force between the sample and the tip.
I mean, most of people think he won Nobel Prize for general relativity, but he didn't.
AFM, atomic force microscopy, STM, scanning tunneling microscopy, and also TM, transmission electron microscopy. They have different methods, they have different ways to realize the microscopic image. The smaller object that you want to observe, the bigger the microscope you need. Next door we have a TM, transmission electron microscope, that is as big as this room. And then the resolution is really small. We have discovered a new type of crystals based on this molecule. Sir Isaac Newton was much more smarter than anyone else in the world of physics.
Okay. Hello everyone, I am Talib. I am PhD student at the University of Rostock in theoretical physics. So today we have very special guest, my colleague and one of the young scientists in our university, Moha Naeimi. Today we're going to talk about organic material science, specifically surface sciences. So welcome, Moha.
Thank you for inviting me.
So can you just tell us about your specialty? What is surface science?
So surface science is one of the subdivisions of solid-state physics. So the science is about studying the effect of surfaces, different surfaces and interfaces. For example, the surface of a solid material or a metal with ambient condition like air or in vacuum. And then the idea is to study the effect of these two interfaces. And by material science, it means that we can study different materials. For example, organic semiconductors, my specialty, or metallic materials, or like water, or any kind of material that you can think of.
Okay, I see. Okay, I have some questions, but before start the science, I have it's wondering more than questions. So, I'm theoretical physicist, so in theoreticians community, they always make some jokes about experimentalists. I mean, it's not like jokes about their knowledge or their integrity, but it's mostly how they do the measurements and how they interpret results.
So what is the joke?
So, I don't know. I can tell you right now.
Why not?
Yeah, you may get offended.
That's not a problem for me. So I'm cool with it.
Yeah. I was wondering, like, what's this situation in experimentalist community? Do they do jokes about theoreticians?
Yeah, actually, there is a joke about theoreticians in experimental world that says there was once a farmer who had a chicken who didn't lay any eggs. So he asked a theoretical physician for help. And the physician did some calculations and he went back to the farmer and said, okay, I have a solution, but it only works for spherical chickens in the vacuum. So that's kind of a joke that we usually have about theory. But of course, in experimental world, we cannot consider the situation as ideal as you do. Yeah. So. And yeah, that's it. But I don't think that these are the jokes. These are like the facts that our differences.
Yes,
so,
yeah, that's right.
Yeah.
Okay, yeah, that's nice. Okay, let me ask my first scientific question about your specialty. So, you work with the molecular crystals, right? You create some crystals with organic molecules. So could you explain what is molecular crystal at all? So could you pick like any molecule and crystallize them?
Yeah, in principle, yes. So any kind of molecule or atom can be crystallized under some conditions, but a molecule crystal means that in one assembly, the molecules are sitting next to each other very regularly. So we can define a unit cell, so a couple of molecules as a pattern, and this pattern repeats entire through the entire crystal or the entire assembly. If this condition is preserved, it means that the material is crystallized. And we have different conditions for crystallizing. For example, one would heat the material to get it crystallized, or one would cool down the material to get it crystallized, like water. And so on. So in general, a crystallized structure is a structure that the molecules are well patterned.
Okay. It's like not atoms, but sitting just as single molecules, right, in the lattice. So
yes,
that's the difference between the.
I mean, you can molecule crystallized material or atomic crystallized material. Doesn't it's not that different in terms of definition, but yeah. So that's it.
I see. Okay. Okay. Then, I mean, why do you crystallize them? I mean, why they are important? So
because we want to get as close as possible to your theoretical ideas. We want to make the world, well ordered, because actually one advantage that the crystallized material can give you is that then you can repeat the properties of one unit cell to the whole assembly. So it's a well ordered structure that you can think of one unit cell or one like a couple of molecules, how they behave, and then repeat this structure. And in our case, we are looking after exciton diffusion in organic semiconductors. And exciton diffusion actually is more pronounced in crystallized materials because they are well ordered, and then they are highly anisotropic. So in one direction, the exciton diffusion is so efficient, and the other direction not. And that would make crystallized structures for us very interesting.
Oh, I see. Okay, just consider like our audition is just the general, and some of them are not specialists in this kind of field. Could you just say tell us about what's the exciton? Just simply.
Exciton actually is an electron hole pair. When you excite some kind of material, it means that the electron gives the excite by the light. It gives the energy from the photon or any other source, and then it goes to another energy level, and then there will be an electron and an empty place of that electron, and this electron and the hole is called an exciton, and then this exciton can diffuse around through some kind of physical processes, or it can decay back and then gives you the photon. Something like that. For example, a fluorescence is a generation of a singlet exciton, and then it decays immediately back. So in simple terms, an exciton is an excited state, where the electron is not in the ground state, but in the excited state.
Okay. Okay, let's back to crystals. So what's the basic procedure to prepare the crystals? I mean, how you start it? Could you say it like in simple terms, like you put the molecules in the pot and heat it, or how?
Not in the pot, of course, but we have some, because we are surface scientists, so we put our material on the surface because we care about the surface. So you can crystallize your structure in some kind of solution, for example, in water, or I don't know, any other solution. But we don't do that. We place our materials on the surface, and we call this surface the substrate. So something that is underneath your system, and then the substrate can be like a graphite or like silicon or glass or any other surface. And then we put our raw material on the surface. I mean, in my case, I put my raw material on the surface, and then I heat it. The key idea is that I heat it really fast so that the temperature could be very high, and the crystallization starts before the material vaporizes and goes away.
Okay.
That's the key idea. And also I add another condition, and I create a micrometer scale environment, a completely enclosed micrometer scale environment, so that when I heat it even above the sublimation temperature of the material, no material loss happens. So all of the material can directly be crystallized very fast, very efficient.
That's cool. Okay. Okay, we both works light matter interactions, the project, right? So I think that in our institute, everyone study light matter interaction, probably. So, I mean, my question is why it's important to study the light interaction with the matter. So what kind of information can we get from this study? Yeah,
it depends on your perspective. I mean, in general terms, light matter interaction is happening all the time.
Yeah.
You know, like when you see stuff, light matter interaction is happening, like in reflection, you know. But in our case, our studies are focused on absorption of the light by the material. And this absorption can create an exciton or create some kind of changes inside the material. And that is that leads to a lot of interesting properties. For example, charge transfer or energy transfer. And you have to know this kind of stuff, how different kind of materials in different shapes, like crystallized, amorphous, or any other phase, how they react to the absorption of the photon. And then you can create, for example, like organic light emitting materials, organic capacitors, I don't know, organic structures that are based on light matter interaction.
I see.
But again, it depends on your perspective. How do you want to look at your this question? I'm sure if you ask someone who is working on quantum optics, they will give you a different answer. But the key answer is that it's very important. Light matter interaction is very important.
Okay. To study the light matter interaction, I mean, your main tool is the laser, right? You always use laser. So just imagine like Albert Einstein didn't create the fundamental law which later used to create the lasers. So you don't have any laser. What would be your tool then?
I don't know. Then I will be doing, I don't know, I would become a DJ then. I would have no idea about this stuff. But with the help of the laser, actually the laser is not our only prop in surface science. We are using actually scanning prop methods with the use of a tip. So, for example, in scanning electron microscope, we are using the electron beam.
STM, right?
SEM, ZEM.
Okay.
And also in STM, scanning tunneling microscope, we are using a conductive tip that is scanning above the surface. And also in AFM, we are using a cantilever that measures the force between the sample and the tip. So laser is not our only prop in surface science we can use. But I'm mostly interested in light matter interaction. But of course, our tools are not limited to light matter interaction. I can do atomic force microscopy or scanning electron microscopy, and also transmission electron microscopy. So the prop is not the photon anymore. There are electrons or the tips.
Okay, I see. Okay. I mean, I know the different type of like the spectra, right? I mean, usually what you get from the experiment, some spectral picture or something, right? Most of the time. So I know there are different types like absorption spectra, emission spectra, or photoluminescence spectra. I don't know the difference between them.
You don't know?
I mean,
Okay.
I know, but maybe you know better. So because you measure it directly, right? So
Yeah, the spectra is nothing but changing one variable
yeah,
and look at the other variable and see how it behaves.
yeah,
That's kind of a spectra. So it's not
yes,
it's a function. You
yeah,
know?
yes,
It's a very pure function what you are measuring, and depends on what kind of measurement you want to do. For example, here in the PEEM lab, we are doing electron spectra.
mm-hmm.
So in terms of photoelectron, we change the we measure the energy of the electrons based on the time. So the variable here is the time, and we look at the number of electrons that we can detect through time. And that is an electron spectra. For example, a photoluminescence spectra is that you detect your photons, and then you measure the energy, and then the variable here is the energy. So you have different energies or different wavelengths, and then you know how many photons are detected at this specific energy or specific wavelengths, and so on. Or absorption spectra, how many photons are absorbed at this particular or this wide range of wavelengths and energies.
Yeah, actually, the principle is the same, but only the difference is just what property you are looking at, right?
What is changing?
Yeah.
What you are changing and what is detected when you change the thing? Yeah.
I see.
In principle, yes.
Yeah. Okay. Okay. Here is the photoemission microscopy, right?
Photoemission electron microscope lab.
Okay.
Yes.
Okay. Could you explain like the basic principle of photoelectric emission microscope? I mean, how you measure it and how you get the electrons from the sample?
Yeah. So the photoemission electron microscope is designed based on the photoelectric effect that Albert Einstein won a Nobel Prize for that, for introducing and explaining that. So it means that when you apply some light to some kind of material, if the photons inside that light are strong enough to kick out one electron from the sample, this is called the photoelectric effect. And then in photoemission electron microscopy, we are interested in the electron that is coming out. And then we capture it, we guide it to our microscope with static electrostatic lenses. And then these electrons can hit the detector, and we can measure all of things. So we can magnify the trajectory of these electrons that are coming out, or we can measure the time in which these electrons hit the detector, and or we can perform electron spectra, and so on. And our prop here, of course, is the laser. So we are using coherent lasers with really narrow bandwidth. And of course, what you are observing is the number of the electrons coming out from the sample.
Okay, yeah, I just want to highlight here. So you told about Albert Einstein. So I mean, most of the people think he won Nobel Prize for general relativity, but he didn't. So
No.
he won for this
He won for the photoelectric
effect.
Yes.
Yeah.
Actually, he modelized the photoelectric effect.
Yeah.
So he invented he realized some kind of equation for that.
Yeah, that's true.
It's the thing that we are measuring here actually is that equation that Einstein introduced. Yeah. Of course, more complicated because Albert Einstein was a theoretical physicist, not an experimental. So.
Okay.
Yeah, actually, and also the photoemission electron microscope was invented by Ernst Bauer, an American German physicist who's right now is 98 years old.
He's alive.
Yes, and he's the inventor of these kind of different microscopes based on photoelectric effect. So he somehow completed the work of Albert Einstein. He has the same reputation as Albert Einstein for us. He's like the god in our
That's
in surface science.
With my background, I'm very far away from experiment. So in my simulations, I can see the atoms, molecules, and the molecular orbitals, atomic orbitals, everything. I can see this, right? So in experiment, how our technologies, how advanced our technologies, can we see single molecule, single atom?
Yes, we can, but not with these kind of devices. So that in molecular resolution, we have AFM, of course, and also STM, and also TM. So AFM, atomic force microscopy, STM, scanning tunneling microscopy, and also TM, transmission electron microscopy. They have different methods. They have different ways to realize the microscopic image. But these are really advanced methods that you can resolve for molecular resolution and also for atomic resolution.
Okay. I think I saw some picture in the papers, like they visualize the molecular orbitals. What kind of microscopy they use to
They are using, I guess, STM. So they place, the work of the STM. How does it work? So the conductive tip approaches the surface, and then you apply some kind of voltage, and this voltage can excite the orbitals, and then based on the excitement of these orbitals, the current changes, and then you can image the current based on your voltage you applied, and then you can image the orbitals. So that was probably STM. And also 2 years ago, I remember we tried to realize molecular resolution with our AFM. So we got a new AFM 2 years ago. It was a very super accurate AFM and also very nice. And we tried, but it was somehow not working. So we were able to see the honeycomb structure of our material, but it was not really reliable. And we realized then for that we need some kind of vacuum environment, because the AFM is doing in ambient condition, like in normal air.
I see.
But also AFM in vacuum is a very powerful device and method for realizing atomic or molecular resolution. These two are really apart from each other. I mean, molecular resolution is completely different from atomic resolution, because for atomic resolution you have to double your technology and this kind of stuff.
Okay. Yeah, then we don't need the very big size of a truck microscopes, right, to see just the molecules. So
actually,
you can see the
smaller object that you want to observe, the bigger the microscope you need. Next door we have a TM, transmission electron microscope, that is as big as this room, and then the resolution is really small.
I see.
You can resolve for a molecular resolution there.
I see.
So yes, so we need a really big microscope for observing these small objects.
Okay. Yeah. What's the coolest tool, experimental tool that used?
So transmission electron microscope, of course, is the coolest. And also I'm using fluorescence lifetime imaging, but the beauty of detecting the electrons is something else. I'm really interested in electron spectroscopy, because electrons can transfer a lot of information with them. And for example, you can measure the dispersion, you can measure the band structure, you can measure the energy. And also it's really nice that you using pulse laser, using packets of photons, like wave packets of photons, you can kick out electrons from your sample, and then you can capture these electrons. It's a very cool method, and that's my main specialty here. So then I would say photoemission electron microscope would be the coolest thing that I've ever done in my entire life.
I see. Okay. What kind of tool that you want to use it in future? I would like to do pump probe. I guess pump probe is a method that is very advanced, and I don't think that it matches the scope of your video, but I will explain it anyway if you want to keep it in your video. But the pump probe is a method that you are using two different light sources, and the pump
actually excites the sample without any photoemission. So the electrons are not out, but it's excited inside the material. And the probe kicks out this electron from the excited state.
I see.
So the idea is that you have two short pulses applying one after each other with a very short time. I mean, I'm talking about picoseconds here.
Yeah, the living time is very short, right?
Yeah, because the excited state lifetime is very short. If you don't probe it, it goes back to the ground state immediately. So you have to probe again immediately after the pulse. I'm talking about picoseconds like that. Yeah. And this is a very cool method. Actually, we have a very nice setup here. It's not complete yet, but I'm seeing myself in the future doing some palm probe measurements. And the other thing would be ARPS, angle resolved photoemission microscopy, which we can do it with our device, but the resolution is not as good as the ARPS. Also, with ARPS, you can detect the band structure of your system, which is really cool method to measure such useful and priceless information from your sample.
Maybe you can tell us about your work. I mean, what kind of molecules?
I'm using organic semiconducting molecules. In principle, anything that contains carbon and hydrogen, I'm interested in. So right now I'm using a very special kind of molecule. It's called rubrene. And this molecule is an organic semiconductor. It's a very well-known, actually, organic semiconductor. It's very famous. And so we have discovered a new type of crystals based on this molecule, which we want to publish the results in a few weeks, I guess. And this new class of crystalline structures based on organic materials can have a lot of applications in organic electronics. For example, in our new crystals that we invented or discovered, I don't know how to call it, we have super anisotropic exciton and charge properties. For example, we realized that using this kind of crystals, we can have crystalline structures with different conductivities. So, in principle, you have one single crystal with different parts with different conductivities, and that might come really useful in organic electronics.
I see.
And also the exciton diffusion is super anisotropic in these kind of crystals. And of course, you can also show some of microscopic images of my crystals in your video. And then, so that's the thing that I'm currently working on. So I want to know the charge properties of these crystals using this device here. And also the optical properties using a fluorescence lifetime imaging microscope. So that's the main thing that I'm doing right now.
Okay, okay, I want to ask about the problems in your work. So, I mean, for my case, like my biggest problem is I don't know, like my computer just freezes sometimes. That's my biggest problem. That's it. So, in your case, I mean, what's the biggest challenge like you face like every day?
Can I first ask you a question? So how do you do when your computer freezes?
I just restart it.
Okay, that's the main thing that we all usually do when we face problems in experimental. So the problem is usually that our setup does not work. The laser is detuned, or the detector is broken, or the vacuum is not working. So these kind of problems are usually the things that we usually face. And especially this device is very complicated. It has a lot of different parts. And for example, one of the problems that I'm facing here is that the laser that excites the sample is not stabilized. So it moves a lot. And then it's very important for us to focus this laser to your sample. And then after one day, the laser goes somewhere else on your sample, and then you don't see your sample anymore in the microscope. And then we are thinking about some kind of beam stabilization setup It's really fancy. So you use position-sensitive diodes to stabilize and then motorize your mirrors that automatically keeps the laser light always at the same position on your sample. And yeah, these are the problems that we usually face. And of course, it's not as simple as restarting the computer, but basically that's the same the thing that we do. So if something is not working, we restart our thinking. So
okay, just imagine I have superpower, so I can buy anything for you. So
thanks.
What would you ask?
I would ask for
Of course, don't ask a car or home. So just
I know. I mean about science. Yeah. So that's a nice question, actually. Yeah, I would ask for an ARPAS first, and a grant that allows me to work on creating a pump probe setup.
I see.
Yeah.
Okay.
And yeah, a lot of stuff, lab equipments. I mean, here we have really high quality lab equipments, but the more quality, the better work. So
my last question would be, there are two scientists in the physics they always put like face to face, Isaac Newton and Albert Einstein. Who would you pick? Which one is better for you?
I don't think that there is one better than the other, to be honest, because they complete their work.
Yeah.
And they are really different in time. I mean, Albert Einstein is so young compared to Sir Isaac Newton. But yeah, I mean, I'm sure Sir Isaac Newton was much more smarter than anyone else in the world of physics.
Okay.
He was super smart.
Yeah. I agree that.
Yeah.
Yeah, it was a delight. So thank you, Moha.
Thank you for inviting.
Informative talk. Yeah.
Great.
Yeah. Thank you so much, and we wish good luck to your work and future plans.
Yeah. Thank you so much.
Thank you.
We probably see each other soon.
Yeah.
1 minute after this.